Great, thank you. Well, I want to really thank the organizers for this invitation for this very unique event, and I'm really looking forward to interacting with you all in this very interesting context. I think we're really at a unique time in sort of human history where maybe the ability to understand and engineer ourselves is reaching really interesting cusps of possibility. And so it's a very timely occasion to think about what does technology, what does science, what does engineering and couplings to things like AI and other topics mean. I direct a group at MIT that works on technologies for trying to understand the brain at a very fundamental level and to try to enable the study and repair of the brain down to its building blocks, molecules, connections, and elementary signals. And so one of the motivating factors for me personally is this idea of making a computer simulation of the brain. Could you simulate a decision or a thought or an emotion in software, and maybe down to molecules? Maybe we don't need that much detail. But one of the driving factors in our thinking is, could you understand how molecules work in cells and cells work in networks and networks work to generate things like behaviors and how they go wrong in pathologies? And so for the last couple of decades, I guess, my interests have mostly been in the realm of technology to create ways of mapping and controlling neural circuitry. But as I'll hope to show today, maybe some of these techniques are ready to be integrated in interesting ways that could be interesting in terms of probing things computationally and increasingly clinically. So today I'll talk about a couple techniques that we've been working on where we're trying to make maps of molecules throughout the brain, be able to control high-speed neural dynamics of the brain and to image the signals of the brain. And then at the end, I hope to convey the idea that maybe we could integrate these tool sets. Imagine imaging many signals at the same time in a living brain, and then be able to causally probe what certain signals might mean, and then at the end of the day to understand the structures that gave rise to those signals. Could you integrate that into some kind of computational simulation of what a brain might do? So let's start with imaging. And in particular, how do you image a structure as large as the brain? Brain cells are gigantic objects, biologically speaking, by far the biggest cells in the body. But the connections between brain cells and the molecules at those connections are very tiny. And so many strategies have been developed for imaging structures with fine precision, super resolution microscopy, cryo-electron microscopy, and so forth have been very foundational. But all of these methods struggle to image large objects like brain circuitry. And so one thing we've been exploring is this idea of taking brain specimens, preserved ones, and making them essentially as arbitrarily large as you want through a physical process of swelling them. So can you take a brain specimen, not a living thing, create a dense mesh of swellable polymer, like sodium polyacrylate, the active ingredient in baby diapers, and then if you anchor molecules to the polymer, loosen up the structure of the brain, and add water, the diaper polymer swells, and you can make the brain larger. And so we call this method expansion microscopy. Panel B here is a piece of the mouse brain, a couple millimeters on a side. And panel C is the same piece of mouse brain tissue, about a day and a half later, let's say, and it's been swollen by about 100 times in volume. And so the polymer mesh starts out very dense, like in the upper left of this cartoon, and it ends up in this extended conformation, like in the lower left. And so both by design, there's not really time to go into all the details, but also through lots of validation, the expansion process is very even. And so this little time-lapse movie just shows the piece of mouse brain tissue being expanded. We formed the polymer earlier, and then we add water right there. And so this is on the order of half an hour, split up to on the order of half a minute. But I hope you can see that this piece of brain tissue is expanding as the water is being absorbed by the baby diaper-ish material. So the basic idea then is to take a brain cell like this golden one on the left of this cartoon and to make it look like the thing on the right. Two biomolecules that we're touching, we pulled apart until there's some minimum distance apart, and two biomolecules that are some larger distance apart will be scaled up in their distance by some linear factor. So it's like you draw a picture on a balloon and you insert air into the balloon, the picture gets bigger, it's the same picture, but all of the ink molecules got separated. And so even from the very beginning, it was very clear that we could use this technique to do kinds of investigations that were very, very difficult to do through earlier means. So on the left-hand side of the slide, we're zooming in from top to bottom. on a piece of the mouse brain. There's a color code on the left. We're decorating certain proteins with fluorescent antibodies. And on the right is the same specimen of the brain and same microscope and everything, but after we've expanded it. And so the fuzzy stuff on the left becomes much more clear on the right. In fact, those purplish dots in the lower left, you can separate into pink and blue components. And those are synaptic densities on different sides of synaptic connections of the brain. And the distance we measure between those protein densities is the same that you would measure with a classical nanoimaging method. This is a little movie of the kind of data that one can get through such investigations. The color code is sort of similar to what we talked about in the previous slide, but you can kind of see that we can get sort of multi-scale understandings of brains because you can zoom in to very, very fine features like individual neural connections, but you can also zoom out and get context of the circuitry. I think I'll skip this slide because I don't think I can click on it with the mouse. I'll give you a little fly through of the fly brain. This is an example here of color coding brain cells, which others have been doing for many years using fluorescent proteins from corals and jellyfish and so forth. You can genetically express them in brain cells. We then can form the expansion polymer and make it bigger. This is a piece of the mouse brain involved with many different kinds of function, including, amongst many things, memory formation. And if you see the top middle of the slide, that's a small piece of the brain zoomed in from the white square below. It's a bit fuzzy because the wiring of the brain is very, very tiny, but the upper right is the same field of view on the same microscope, but after we've swollen it, and now we can see the wiring of the brain much more precisely. You can iterate this expansion process. So we can form the polymer and then expand a piece of brain tissue and form a second polymer in the space opened up by the first expansion and expand it again. And so you can kind of do this over and over again. So if you do it 2 times, you can start to see very, very fine structures, including even parts of individual synapses. So synapses are not uniform things. They're micro machines with incredible amounts of molecular content organized very precisely in 3D. And so we were able to look at these very fine structures and look at the alignment of presynaptic proteins and postsynaptic proteins, which others have characterized in cultured cells, but which have been difficult to look at in intact tissue with this kind of precision. And we're able to map out the alignment of interesting molecules like presynaptic calcium channels and postsynaptic scaffolding proteins. So a lot of people are doing this method of expansion. The fundamental building blocks of life, biomolecules are nanoscale, and if you're trying to understand how they work together in a system, that means seeing how they look and how they relate to each other in 3D in the context of a system. So not surprisingly, a lot of people started to use it, not just in neuroscience, but in other parts of biology. This slide really isn't meant to be read, but it sort of dramatizes the idea that there's a lot of demand for nanoimaging. And importantly, we've really tried to democratize nanoimaging. The techniques are very easy to use, and ease of use is a big priority for us as we develop techniques. We want to build tools that don't require elaborate hardware or extraordinary skill. We used to have lots of hands-on workshops for this kind of thing, but when COVID broke out, we resorted to doing photographic tutorials, which have been in use by many people in order to do this kind of nanoimaging. So to summarize this first of the short stories I want to talk about today, we've been able to physically magnify biological objects in an even way. And this allows for the mapping of molecules and brain wiring and other kinds of things at scale. And so it's very intriguing to think about whether you could make a brain circuit map that's detailed enough to look at, for example, the underlying mechanisms through which a memory is encoded or through which the circuitry that is organized underlying things like emotions or decision-making is architected. Now, the obvious limitation of expansion is that it's probably never going to work on a living thing. And so in the next part of the talk, I want to focus on various tools for the imaging and control of living brain dynamics. And I think I'll just briefly talk about optogenetics, because it's a fairly, at this point, established technique. But the basic idea is simply to use light to control brain activity. We borrow molecules from the natural world that convert light into electrical signals and put them into brain cells through genetic delivery mechanisms that are delivering the genes that encode for those molecules. Light can be aimed at brain cells and coupled to those molecules to transport charged particles into and out of the brain cells. And then we can bring, just as electrodes are routinely brought into the brain, we can bring light into the brain through optical probes. And so the real serendipity and sort of underlying luck and miracle of all this is that the molecules that we use are from the natural world. They're light-driven ion channels and pumps that are found in single-celled microbes and creatures all over the tree of life. And they look a lot like this molecule in the lower left. This is a membrane protein that contains a vitamin A variant, all transretinal. When hit by light, this molecule that sits in the membrane of single-celled microbes will translocate ions from one side of the membrane to the other, converting solar energy into chemical energy. And so the first to be found was a light-driven proton pump found in single-celled archaea. Later, people found light-driven chloride pumps and pumps of other kinds of ions. And later, people found light-driven ion channels that convey various kinds of ions across cell membranes. And so what we've been doing over these years is identifying molecules that are safe enough, fast enough, powerful enough that you could express the gene that encodes for these proteins in brain cells, shine light on the brain cells and activate them and shut them down. And so one of the exciting things about the strategy is that we can use just a simple gene that's delivered into the brain. The brain cells will then manufacture the protein, and then we can shine light at the appropriate color and then turn the brain cells off in the case of translocating charged particles in the proper direction on the left in the middle columns of the slide, or you can bring charged particles into the cell through a channel that's shown on the right-hand part of the slide. And so by putting positive charge into a cell, we can activate it, you can trigger a brain cell and figure out what kinds of events you can initiate or sustain. And by turning cells off, you can figure out whether they're necessary for a given function or not. And so these are pretty widespread tools in use. And one of the things that's been exciting is people are starting to apply these tools in different and interesting contexts throughout biology ever since the molecules were presented, because again, they're very easy to use. You can put the gene into the cell and shine light on them. And so I'll just maybe mention one example, kind of riffing off of Wolf's introduction of oscillations, led by my collaborator Li Wei Sai, where they're driving oscillations in the brain at a certain frequency in Alzheimer's model mice. These are mice engineered with a gene that provokes Alzheimer's-like symptoms. And when they drive the brain cells at this specific frequency of 40 times a second, then the molecular pathology in these mice gets cleaned up a bit. And later the teams went on with Li Wei's leadership to work on ways to deliver these brain waves through non-invasive means like a flickering light that the mice would watch. And now we're working on human trials of this. We're basically having people with Alzheimer's watch a movie, trying to see if they can benefit from the flickering light. And those are trials being both conducted in a startup company that's trying to do more scalable trials that we started, as well as in academia. You can try to make the precision of the technology more accurate in spatial dimension. So in work that's being led by people like Valentin Emiliani and others, people are working on projectors that can sculpt light, so you can enter information into individual cells very precisely. You can then activate cells in certain patterns and then elicit sequences and other kinds of interesting 3D coordinated patterns of activity in the brain. And then I'm sure Boton Ruska will talk about this more, but recently, of course, these molecules, which have up till date been primarily used in animals to study the brain, are starting to be explored in humans as well. So could you use these in humans who are needing sensory restoration and potentially other things down the road in order to improve the human condition? So I think I'll just maybe end there because I want to talk about some newer things that have just been coming up in quite recent years. But one of the things that we're very interested in now is in imaging brain signal. There's so many kinds of signal in the brain. How can we understand all of them, ideally, or a fraction of them that you could infer fundamental mechanisms of how cells signal in the brain at a molecular level? Let's begin with one note, which is, we have optogenetic molecules where we can control brain cell electrical activity with light. You can take these molecules, and several groups have been able to mutate them in order to make them run in reverse. You can use them to image brain activity. They become fluorescent indicators of neural voltage. And so we've been working in that space as well. This is some work that we collected in collaboration with Shahan's group, the EU, where you can express one of these now fluorescent molecules in a brain cell using gene delivery, shine light of one color on the brain and image with the second color of light and read out neural activity without an electrode. You can do it with a camera now. And so it's interesting to think about whether you can now image many cells at once and look at their neural activity in a non-contact way, and also to use a second color of light, as we talked about before, to control brain activity. So this is very intriguing, the idea that you can use light both to read and write information to the brain. And if the optogenetic molecules are starting to be explored in the human body, then one can imagine these neural readout molecules, which are mutated optogenetic molecules, being potentially also used in neural prosthetic application. But voltage is just one of the many, many cells in the brain, signals in the brain that cells use to compute. I mean, if you think about almost any brain function, learning, the acquisition of a language, the practice of a skilled movement, the progression of aging, or the development of the brain, these all involve processes that take place over very extended periods of time, months, sometimes years. And so this is just a little cartoon borrowed from Wikipedia of a cell which has on the left some inputs coming into it, nutrients, hormones, you name it. On the right, the cell might turn on some genes in the nucleus. It might live or it might die. It might move or it might stop moving. And in between, of course, are who knows how many, but certainly in the hundreds of thousands of gene products that encode in a computational way the transformation of short-term stimuli into long-term changes. And so a lot of biology is really being worked out as interface between cells, the molecules that make them up, and the emergent properties of what the cells generate in network form. So decoding these signaling networks in cells is very daunting. And these little circles represent different biomolecules, and the little arrows represent sometimes an entire PhD's worth of work just to decode one of these arrows. And of course, figuring out what they mean in a given cell or context is very tricky. So we started wondering several years back, well, what if we could just image many of these at the same time? So people have tried to make indicators by taking a fluorescent protein from corals or jellyfish and fusing it to some kind of sensing domain. But if you want to use many of these at once, it's difficult, right? Maybe you have to make one that's a color like green and a second one that's a color like red. But what if you want to see 3 or 4 or 5 or an arbitrary number of such signals? So we started to think about whether we could use engineering ideas implemented through chemistry in order to image many, many things, potentially an arbitrary number of things in a living cell. And I'll just talk about one of these methods, spatial multiplexing. And the basic idea is to take these fluorescent indicators of different signals and park them at different places in the cell. So in this cartoon, suppose that the spots labeled 1 could have indicators of one kind of cellular signal, like calcium, and the points labeled 2 could have indicators of a second signal, like, for example, protein kinase A. And of course, there might be hundreds of such signals involved with a given event, like a learning event, or the conversion of a neural injury into some kind of inflammatory state, or other functions or dysfunctions. So while the cell's alive, these little points, which ideally are close enough that they can sample the biology you care about, but far enough apart that you can resolve them, these points will blink out little movies of the physiology that you're seeing there. And then when you're done with the experiment, you could preserve the cell or the brain and then label those little puncta of indicator to figure out which one was where. And so here's just one way that we came up with doing it. You can take different fluorescent reporters, fuse them to different self-assembling peptides, and then those peptides will cluster these indicators at different points in space and enable them to stably and safely, biologically sample the physiology happening at those different points. So these self-assembling peptides are human-designed proteins, but they act sort of like the coat proteins of viruses. They self-assemble into clusters. And these constructs also bear a little epitope, which is basically something you can stain for to figure out what signals were where later. And so in panel B here is a cultured neuron with a calcium indicator, GCAMP6, a genetically encoded indicator made by others. And in panel C, we've taken that indicator and fused it to self-assembling peptides, and they cluster at discrete, random, but stable points throughout the neuron. And we did lots of control experiments to check out changes, to look for any potential effects on cell health or cell physiology or sensor quality, and didn't see any. And so we're very excited that this might be a way to sample biology by parking these indicators at different places in a cell. But importantly, to use multiple indicators at the same time. So here are 3 indicators made by other groups, one for calcium, one for cyclic AMP, and one for protein kinase A. We can park them at different places in a cell through this method of fusing them to different self-assembling peptides and expressing them in brain cells. The green images are what you'd see when the cell's alive. Of course, they'd be blinking out little movies of what they're doing. And the false color images are what you would get after you stained them after the fact. And you can then figure out which sensor was where. And so we can measure many things at once in the same cell. And that's important because then you can see relationships between them. If you image one signal in one cell and a second signal in a second cell, you might miss out on whether they have some kind of relationship where one goes high and the other goes low, or a more complicated relationship. So we can start to look at how the timing, for example, of calcium could relate to the amplitude of protein kinase A. And it's probably not time to go into all the details of that, but we're very interested now in developing all sorts of strategies to extract these kinds of patterns in automated ways. You can add a fourth indicator. Here we threw in a protein kinase C indicator, or we could add in a fifth indicator. Here we threw in an ERK kinase indicator. So to summarize the second part of the talk, we've been working on basically engineering all sorts of strategies for using many fluorescent indicators, mostly made by other people, to use them at the same time so we can try to map out signaling in a cell in the brain. And that complements earlier work that we and others have done with optogenetic control, where ideally you could look at a lot of signals at the same time while the brain is alive, and then use causal perturbative strategies like optogenetics to test whether a certain cell or node in the network is important for that function. So just one last thought before acknowledging the people who have led different projects along the way, which is that we've shared all these tools very freely. The expansion strategies and the optogenetic tools are, I would estimate, in use by over 1,000 groups each already. The live imaging tools are more recent, and so they're just starting to make their way more broadly into the world of biology. But one thing we're very excited about is to try to integrate these tools together. What if you could image many signals in the brain while the brain is alive, perturb brain cells with optogenetics, and then use the expansion method to make maps of how molecules in cells and cells and networks are organized? Could you synthesize these into some kind of computational model? And so we're now working with many others to try to build networks of groups that would work on some small brains, like the worm C. elegans, which has only 302 neurons, or the larval zebrafish. Here we have an image of one with, they have about 100,000 brain cells. This is some work we've done where we were expanding larval zebrafish brains so we can make more detailed maps of it. We can also expand the worm C. elegans. And we can take the indicators I mentioned earlier, like the fluorescent voltage indicators, and also adapt them to fish and to worms. And so it's very exciting to think about whether we could, not just share such tools individually, but to think of them as an integrated pipeline for the performance of such analyses from molecule to cell, cell to network, and network to behavior. So I think I wanted to just maybe spend the last second or two acknowledging all of the people who've led different projects along the way. So the initial expansion work was led by two then graduate students, Fei Chen and Paul Tilburg. And the adaptation to looking at larger scale circuitry with light sheet imaging was led by Reigao Shoasano and Gokul Padiyula, working with Eric Betzig's group and our group. Deblina Sarkar, Jinyan Kang, and Oswasi have led the most recent version of our method of iteratively expanding objects over and over again, based upon initial work by J.B. Chang in the group. The optogenetic work was done with a large set of collaborators and alumni and colleagues over the years. Brian Chow, Xue Han, Amy Chuang, initial work that I did collaborate with Carl Deiseroth, Nathan Klepetke, Or Shemesh, and others. The work that on Alzheimer's disease was led by Li Wei Sai, work done by Hannah Yakurito and Annabelle Singer. Or Shemesh and our group worked with the Valentina Miliani group to work on holographic control. And then the work on voltage imaging was done by Kiro Pijakovic in our group and Erika Jung. And then finally, the work on imaging many signals at once through spatial multiplexing was done by Shannon Johnson and Chengyang Lengu when they worked with me. Of course, there's a much larger list of people who helped with such techniques. I should mention Limor Freifeld doing expansion of the zebrafish brain, and Jay Yu on the expansion of C. elegans, and a very long list of collaborators who've helped make all these things possible. I think that I'll end.